Okay, Bismillahirrahmanirrahim. Hello, everyone. Our today's topic is neurotransmitters. According to neurotransmitters, our learning objectives are, we'll be discussing about introduction and definition of neurotransmitters, criteria for neurotransmitters, classification of neurotransmitters, properties of neurotransmitters, type of neurotransmitters, and finally, disorders associated with neurotransmitters. As far as the introduction is concerned, neurotransmitters are chemical messengers that transmit signals from a neuron to a target cell across a synapse. Target cell may be neuron or some other kind of cell like a muscle or gland cells. Necessary for rapid communication in the synapse, neurotransmitters are packaged into synaptic vesicles, presynaptic side of the synapse. You can look right here, the schematic representation of the synaptic, and you can see right axon, that is of the presynaptic knob containing vesicle, which contain the neurotransmitters, and in between the two, that is the post and presynaptic knob, there is a synaptic cleft, and on top of the postsynaptic knob, there are the receptors. So the post one is called the receiving neuron. So the criteria for neurotransmitters, nowadays, many substances are categorized as neurotransmitters. To consider a substance as neurotransmitter, it should fulfill certain criteria like the substance must be found in neuron. This is the first condition. It must be produced by a neuron. It must be released by a neuron. And after release, it must act on a target area and produce some biological effect. And finally, after the action, it must be inactivated. As far as the classification of neurotransmitter is concerned, depending upon the chemical nature, neurotransmitters are amino acids. The neurotransmitters of this group are involved in fast synaptic transmission and are inhibitory or excitatory in action. For example, GABA, gamma-aminobutyric acid, glycine, glutamate, and aspartate. The second group is amines. Amines are the modified amino acids. The neurotransmitters of this group involve in slow synaptic transmission and are inhibitory and excitatory in action. For example, noradrenaline, adrenaline, dopamine, serotonin, and histamine. Others, the ones which do not fit in any of these categories, the examples are acetylcholine and nitric oxide. Depending upon function, the neurotransmitters are grouped into excitatory neurotransmitters, in which it is a chemical substance which is responsible for the conduction of impulses from presynaptic neuron to postsynaptic neurons. The neurotransmitters released from the presynaptic axon terminal does not cause development of action potential in the postsynaptic neuron. Rather, it causes some changes in the resting membrane potential, slightly depolarization by the opening of sodium channels in the postsynaptic membrane, and the influx of sodium ions from the extracellular fluid. The slight depolarization is called excitatory postsynaptic potential. Excitatory postsynaptic potential, in turn, causes development of action potential in the initial segment of the axon of the postsynaptic neuron. The common excitatory neurotransmitters are acetylcholine and noradrenaline. The second class is inhibitory neurotransmitters. It is the chemical substance which inhibits the conduction of impulses from the presynaptic neuron to the postsynaptic neuron. When it is released from the presynaptic axon terminal, due to the arrival of action potential, it causes opening of potassium channel in the postsynaptic membrane and a flux of potassium ions. This leads to hyperpolarization, which is called inhibitory postsynaptic potential. When inhibitory postsynaptic potential is developed, the action potential is not generated in the postsynaptic neuron. The common inhibitory neurotransmitters are GABA, gamma-aminobutyric acid, and dopamine. Transport and release of neurotransmitters. The neurotransmitters is produced in the cell body of the neuron and is transmitted through the axon. At the axon terminal, the neurotransmitter is stored in small packets called vesicles. You can see right here. These are the small packets called the vesicles which control the neurotransmitters. At the axon terminal, the neurotransmitter is stored in small packets called the vesicles. They are right here. Under the influence of stimuli, these vesicles open and release the neurotransmitter into the synaptic cleft. You can see right here. This is synaptic cleft, and they open up and release the neurotransmitter into the synaptic cleft. Then in the synaptic cleft, it binds to specific receptors which are present on this upper membrane of the postsynaptic knob. Right here, you can see right here, these blue ones. On the surface of the postsynaptic cell. So they bind to specific receptors which are present on the surface of the postsynaptic knob. The receptors, G protein kinase, or ligand-gated receptors. Inactivation of neurotransmitters. After the execution of the action, neurotransmitter is inactivated by 4 mechanisms. After performing its function, the neurotransmitter is inactivated by 4 mechanisms. Number 1, it diffuses out of the synaptic cleft to the area where it has no action, or it is destroyed or disintegrated by specific enzymes, or it is engulfed and removed by astrocytes, and finally, it is removed by means of reuptake into the axon terminal. Reuptake of neurotransmitters. Reuptake is a process by which the neurotransmitter is taken back from synaptic cleft into the axon terminal after execution of its action. The reuptake process involves a specific carrier protein for each neurotransmitter. As far as the properties of neurotransmitters are concerned, number 1, they are synthesized in the presynaptic neuron, localized in vesicles in the presynaptic neuron, released from the presynaptic neuron under physiological conditions, rapidly removed from the synaptic cleft by uptake or degradation, presence of receptor on the postsynaptic neuron, binding to the receptor elicits a biological response. You can look right here. This shows the whole process in the form of diagram. You can see right here. This is the axon containing, there is a mitochondria in there. In mitochondria, the acetyl-CoA enzyme comes out, binds to the choline and makes acetylcholine. The acetylcholine is present in the synaptic vesicle. You can see right here, it's present in the synaptic vesicle. This acetylcholine then goes and binds to the receptors present on the postsynaptic vesicle, so postsynaptic membrane, and then from there it is again broken down into choline and acetate. Choline goes back into the axon again, and that is the nerve terminal of the axon again, and combines with the acetyl coenzyme A, makes acetyl-CoA and makes a choline, combines with the choline and makes acetylcholine, and this process keeps on going. So this diagram actually showing the events of cholinergic axon terminal. Types of neurotransmitters. They are divided into two main classes, but there is a third one also. Excitatory, inhibitory, and the ones which have both. That is excitatory as well as the inhibitory types or inhibitory function. Excitatory include glutamate, aspartate, and nitric oxide. Inhibitory, glycine, GABA, serotonin, and dopamine. And both contains acetylcholine and norepinephrine. Classes of neurotransmitter, as I pointed out earlier, there may be amino acids, amines, monoamines, soluble gases, and the acetylcholine. Amino acids include glutamate, aspartate, glycine, and GABA. Monoamines, catecholamines, that is dopamine, epinephrine, and norepinephrine. The other one, indolamines, like serotonin. Soluble gases, nitric oxide and carbon monoxide. Acetylcholine is the acetylcholine. Coming to precisely about serotonin, which is 5-hydroxytryptamine, synthesized in 2 steps from amino acid tryptophan, regulates attention and other complex cognitive functions such as sleep, eating, mood, and pain regulation. Too little serotonin has been shown to lead to depression, anger, and anger control, and so on. As far as the biosynthesis is concerned, starting with the tryptophan, by the action of the enzyme tryptophan hydroxylase, it is converted into 5-hydroxytryptophan, and by the action of 5-hydroxytryptophan decarboxylase, it is converted into 5-hydroxytryptamine, which is a serotonin. The processes of neurotransmitter action, there are 7 processes by which a neurotransmitter can act or perform its function. Number 1, you can write here, neurotransmitters are synthesized from precursors under the influence of enzymes. You can see right here, the synthesizing enzymes, neurotransmitter precursor, these are neurotransmitter precursors. So by their action, the neurotransmitters are synthesized from precursors by the action of enzymes. Number 2, after synthesis, they are stored in vesicles. You can see right here. Neurotransmitter is stored in the vesicle. Number 3, neurotransmitter molecules that leak from their vesicles are destroyed by enzymes. They are present in the vesicles, and somehow if they are leaked, then they are destroyed by the enzyme, that is the degrading enzymes. Number 4, action potential cause vesicles to fuse with synapse and release neurotransmitters. Action potential causes vesicles to fuse, you can see right here. Vesicles to fuse right here, this one, and fuse with synapse, this is the synapse, and release neurotransmitters, you can see right here. The neurotransmitter is released into the synaptic cleft. Some of it binds with the autoreceptor, you can see right here, This is an autoreceptor, and this is a neurotransmitter, bind to the autoreceptor, and inhibit subsequent neurotransmitter release. Once it binds, then it inhibits the further neurotransmitter release. And finally, released neurotransmitters are deactivated either by reuptake or enzyme degradation. These released neurotransmitters, they are then deactivated either by enzyme or by the reuptake or by the enzymes. Steps in neurotransmitter processing are, number one, synthesis. Neurotransmitters are synthesized by the enzymatic transformation of precursors, as you have seen earlier. Then the second step is storage. They are packaged inside the synaptic vesicles. Then they are released from there. They are released from presynaptic terminal by exocytosis when calcium enters axon terminal during an action potential. Then it diffuses across the synaptic cleft to the postsynaptic membrane, where in the postsynaptic membrane, they bind to the receptor proteins which are present at the surface of the postsynaptic membrane, and the neurotransmitter is degraded then either by being broken down enzymatically or reused by active reuptake. Diseases associated with neurotransmitters. Neurotransmitters, for example, acetylcholine causes the disease Alzheimer's. Dopamine, responsible for Parkinson's and schizophrenia. GABA, for epilepsy. Serotonin, migraines, and ADD. That is attention deficit disorder. Attention deficit disorder and depression. Glutamate, responsible for migraine and stroke. So you have to remember these diseases related to the neurotransmitters. Small molecule neurotransmitters, as I pointed out earlier, they are acetylcholine, which has the excitatory, amino acids, gamma-aminobutyric acid, which has the inhibitory effect, glycine, inhibitory, glutamate, excitatory, aspartate, excitatory, and biogenic amines like dopamine, inhibitory, noradrenaline, excitatory, serotonin, inhibitory, and histamine, excitatory. Histamine, amino acid histidine is the precursor of an important neurotransmitter, histamine. Histamine is present in venom and other stinging secretions. You can look right here. This is histidine. By the action of the histidine, decarboxylase is converted into histamine. And the enzyme histidine decarboxylase requires pyridoxal phosphate. So by the removal of carbon dioxide, histidine is converted into histamine. Histamine generally it does not penetrate the blood-brain barrier and hence must be synthesized. Because it cannot penetrate, so it must be synthesized there. So histidine, in the presence of histidine decarboxylase and pyridoxal phosphate in the present neuron, is converted into histamine, which releases and then goes to the astrocytes. Histamine is a biogenic amine involved in local immune responses, regulate physiological function in the gut, act as a neurotransmitter, triggers inflammatory response. GABA, gamma-aminobutyric acid, synthesized directly from glutamate. GABA is the most important inhibitory neurotransmitter. Present in high concentration in the central nervous system, preventing the brain from becoming overexcited. If gamma is lacking in certain parts of the brain, epilepsy results. And this is the structure of GABA. There's gamma aminobutyric acid. You can see right here the alpha carbon, beta carbon, and the gamma carbon. Carbon next to the carboxylic is known as alpha. Next one is beta, and the third one is the gamma. So it's an amino acid present at the gamma carbon, so it's known as the gamma aminobutyric acid. Synthesis, storage, and function. It is synthesized from glutamate by glutamate decarboxylase. Right here, you can see right here. And this is the carbon dioxide which is released, butyric acid, and this is the carbon dioxide. You can see right here. This is a glutamic acid. Glutamic acid is dicarboxylic acid. One carboxyl is attached to the alpha carbon, also attached, and which is also attached to another carbon, and there is a extra carboxyl group. So this, by the action of enzyme glutamate decarboxylase, the enzyme next to amino group is this one present in the alpha carbon is removed, and we get the formation of gamma aminobutyric acid. You can see right here, amino group, carboxyl group is removed, and we get the gamma aminobutyric acid. Gamma aminobutyric acid can also be synthesized through other steps, that is glutamine, by the action of an enzyme, glutaminase, converted into glutamate, then glutamate, gamma amino decarboxylase, glutamate decarboxylase is converted into GABA, and GABA, by the action of an enzyme GABA transaminase, is converted into succinyl semialdehyde, and then that leads to the succinic acid. The action of gamma transaminase is inhibited by vigabatrin. GABA acts as the inhibitory synapse in the brain by binding to specific transmembrane receptor in the plasma membrane of both pre and post synaptic neuronal processes. It is most highly concentrated in the substantia nigra and globus pallidus nuclei of the basal ganglia, followed by the hypothalamus, the periaqueductal gray matter. Synthesis of GABA, as I pointed out earlier, from glutamine to glutamic acid, and from glutamic acid to GABA. Then from glutamic acid to, it is also converted into glutaric acid, which is then converted into succinic semialdehyde, and that converted into succinic acid. Glutamic acid, in the presence of the enzyme GABA transaminase, converted into glutaric acid, and GABA, in the presence of GABA transaminase, also converted into succinic semialdehyde. So glutamic acid and GABA both can be converted into succinic semialdehyde through different channels. GABA receptors. There are two types of GABA receptors. GABA exerts its effect via two types of receptors, GABA-A and GABA-B receptors. Both have certain functions. GABA-A is the endogenous compound that causes GABA-A receptor to open up. So when the GABA binds with the receptor, then causes the GABA-A receptor to open up. And once bound to the GABA, the protein receptor changes the conformation within the membrane so that it opens up the pore in order to allow chloride ions to pass down their electrochemical gradient. Again, in the presence of GABA, bind to the membrane. Because of the binding to the membrane by the GABA, changes the conformation within the membrane. As a result, the pores are opened up that allow chloride ions to pass down the electrochemical gradient. More precisely, in case of A receptor, the presence of GABA increases chloride ion conductance into the cell. This increased chloride ion influx results in membrane hyperpolarization, and as a result, neuronal excitability is reduced and GABA passed into the pathways, into their respective pathways. Look right here. GABA binds to the receptor that opens up the pores in the membrane, and as a result, you can see right here, the chloride pass on from the extracellular to the intracellular membrane and exerts its effect. GABA-B receptors. Activation of presynaptic GABA-B receptors decreases the release of GABA and of different transmitters. Activation of postsynaptic GABA-B receptors modulates the opening of potassium channels, which induce the intracellular potassium exit and consequently a membrane hyperpolarization. GABA-A receptors perform their function through chloride ions, and GABA-B perform their function through the potassium channels. GABA functions: relieving anxiety, improving mood, relieving premenstrual syndrome, regulating the release of sex hormones. GABA deficiency symptoms: anxiety and depression, general uneasy feeling, one can't sit still for long period of time. Health problems that can be caused by GABA deficiency are insomnia, depression, mood disorder, excessive stress, hypertension, atherosclerosis, motion sickness, low level of digestive enzymes, ADHD, that is attention deficit hyperactivity disorder, epileptic seizures, panic disorders, bronchitis, and growth hormone stimulation. This is the diagram that shows the whole mechanism. You can see right here of the GABA. Glutamate, which is present in the presynaptic neuron, nerve terminal neuron. Glutamate converted into glutamic acid decarboxylase into GABA. And GABA then are stored in the vesicles right here. and these vesicles then fuse with the membrane and release into the presynaptic cleft, into the synaptic cleft. And the receptors which are present on the postsynaptic membrane of the postsynaptic cleft, postsynaptic neuron, binds to the GABA, and then they perform its function, and some released ones are then transferred by the gamma transporter into the astrocyte, where GABA is converted back to glutamate and to glutamine. And glutamine is then again carried to the back to the nerve terminal, and this cycle keeps on repeating. Termination of the action, reuptake into presynaptic terminals and or surrounding glial cells via reuptake transporters. Function, inhibitory neurotransmitter in the brain, cerebral Purkinje neurons, cerebral cortex, substantia nigra, globus pallidus, and so on. 20% of CNS neurons are GABAergic. 30% of all the synapses, presynaptic inhibition within the spinal cord. Finally, in conclusion, GABA is an amino acid made in the brain cells from glutamate. It functions as an inhibitory neurotransmitter, meaning it blocks nerve impulses. Without GABA, nerve cells fire too often and too easily. Coming to acetylcholine. Acetylcholine is synthesized in nerve terminals from acetyl coenzyme A, which is synthesized from glucose, and choline in a reaction catalyzed by choline acetyltransferase is converted into acetylcholine. The presence of choline acetyltransferase in neurons is thus a strong indication that acetylcholine is used as one of its transmitters. Acetylcholine is secreted by neurons in many areas of the nervous system, specifically by the terminals of the large pyramidal cells from the motor cortex, several different types of neurons in the basal ganglia, the motor neurons that innervates the skeletal muscles, the preganglionic neurons of the autonomic nervous system, the postganglionic neurons of the parasympathetic nervous system, some of the postganglionic neurons of the sympathetic nervous system. Acetylcholine is the transmitter of the parasympathetic autonomic nervous system and of the sympathetic ganglia. Stimulation of the parasympathetic system produces effects that are broadly opposite to those of the sympathetic system. That is, slowing of the heart rate, bronchoconstriction, stimulation of the intestinal smooth muscle. Acetylcholine also acts at neuromuscular junctions. Acetylcholine may be involved in learning and memory, as neurons containing this transmitter also exist in the brain. This shows the diagram shows the events of cholinergic axon terminal. You can see right here, axon. This we have already shown this diagram before. The mitochondria present there. because within the mitochondria, the TCA cycle takes place and synthesize the acetyl coenzyme A from pyruvate. Acetyl coenzyme A then combines with the choline and makes the acetylcholine, that which is stored in the vesicle, which are present in the synaptic synapse and known as the synaptic vesicles. Then acetylcholine, then from the synaptic vesicle, goes to the released and binds to the receptor present on the postsynaptic membrane. And then some of that goes to the other part, and the choline is broken down into acetate and choline. Choline again goes back, and this cycle keeps on repeating. Synthesis of acetylcholine, this is synthesized from serine by serine decarboxylase, converted into choline, and that choline then is methylated by choline and methyltransferase in the presence of 5-adenosyl-methionine. Remember that S-adenosyl-methionine is the universal methyl group donor. This process keeps on repeating until it becomes the quaternary amine. And this acetylcholine, this choline, which is synthesized, combined with the acetyl coenzyme A, choline acetyltransferase is the enzyme in the cytosol and converted into acetylcholine, which is transported into through the vesicles. There's another diagram showing the whole mechanism. That is choline combined with the acetyl coenzyme A, choline acetyltransferase, and converted into acetylcholine. Dietary choline comes from eggs, soya, liver, and so on. Also enters into from extra to the intracellular space, and intracellular esterases breaks this into acetate and choline. Or it can also be goes to the other channel where the choline transporters are there, and it is carried through the transporter to the this one, and form the acetylcholine. The acetylcholine, which is present in the vesicles, out goes to the extracellular in the form of choline, acetylcholine, which is acetylcholinesterase right here. Acetylcholinesterase breaks it into acetate and choline, and this process keeps on repeating. There's another diagram that shows how it works. Glucose converted to acetyl coenzyme A combines with the choline, removal of the CoA, choline acetyltransferase makes the acetylcholine. Acetylcholinesterase in the synaptic cleft, converted into acetate and choline. Serine, from which acetylcholine it is synthesized, serine decarboxylase into ethanolamine, then it converted into choline in the presence of S-adenosyl-methionine, and it is converted into choline. Methionine can also be synthesized from homocysteine in the presence of vitamin B12. Methionine and methionine then converted into S-adenosyl-methionine and acts as a methyl group donor. Acetylcholine receptors, two main classes of acetylcholine receptors: nicotinic and muscarinic. Nicotinic receptors are inotropic, binds nicotine, found in ganglia and at the neuromuscular junctions. Binding of acetylcholine opens the channel for both sodium and potassium, to pass through the membrane. Action of the ligand on the channel is direct, so action is rapid. Finally, in order to summarize, acetylcholine is a transmitter that transmits signals from one neuron to another or from neuron to muscle to produce action potential. Acetylcholine was the first neurotransmitter to be identified, and its old name was sagostov. The type of receptors that acetylcholine act on is cholinereceptors, muscarinic, nicotinic. Muscarinic and nicotinic. Acetylcholine is synthesized at axon terminal by acetylcholinesterase and hydrolyzed by cholinesterase, and both are present in neuron terminal. Acetylcholine is the substance of nervous system that activate skeletal muscle. Myasthenia gravis is a popular autoimmune disease occurred due to the antibodies directed against acetylcholine receptors and cause muscle weakness and many other symptoms. Alzheimer's disease is very popular age-related neurodegenerative disease, cause loss of recent memory and disorientation and loss of motivation. It related also to deficiency in the acetylcholine. Blocking or mimicking the action of acetylcholine have many uses in medicine. Drugs that acting on acetylcholine receptors, either agonist or antagonist. Blocking the release of acetylcholine at neuromuscular junction by botulinum toxin have a wide range of uses in medicine, especially in plastic surgery and many other uses. Thank you.